Hello, my name is Rebecca Thomas and I'm here with my work colleague Elizabeth Sollars. We're from the Sheffield Children's NHS Foundation Trust. We're going to talk to you about a couple of research projects. So I'm going to talk to you about the proof of principle assessment of the next generation sequencing workflow for rapid newborn screening, which is a research project funded by the Health Innovation Challenge Fund. And Lizzie's going to talk to you about cystic fibrosis in newborn screening. So newborn screening in the UK is organised and supported as part of the NHS. We've got 16 screening labs nationwide, which test between 30,000 and 130,000 babies each per year. Screening is performed free at the point of care to the family, and the process is governed by agreed national targets and the Newborn Screening Committee. So in England, newborns are screened for 9 rare but serious health conditions using dry blood spots. These include sickle cell disease, cystic fibrosis, congenital hypothyroidism, and 6 inherited metabolic diseases. So we wanted to ask ourselves, can a next generation sequencing protocol be optimised for a high throughput capacity and a fast turnaround time? This could potentially be used for disorders where there's no biochemical marker suitable for newborn screening, and it could also be a test that allows sampling shortly after delivery before the baby leaves hospital. So at the moment, birth obviously happens on day 0, and a dry blood spot's taken by the midwife on day 5, and this is when biochemicals stabilise in the blood. A positive result can be obtained by day 7, and so clinical intervention happens no earlier than day 7. With early genetic testing, we can take the dry blood spot the day the baby's born, so this sort of allows increased sort of flexibility with that procedure. So this is just an overview of our semi-automated workflow, which I'll go into detail afterwards. So it consists of punching a small disc from the dry blood spot, followed by DNA extraction, library preparation, library QC, loading the chip onto the Iron Chef, followed by sequencing and bioinformatic analysis. So we punch a dry blood spot using the Panthera dry blood spot puncher at our Sheffield newborn screening facility. We use the 6mm punch head, and punching time takes less than 60 minutes per 96 well barcoded plate. DNA extraction. So this process was optimised using healthy control venous blood and dry blood spot samples. We developed a programme on this biomech robot, and this allowed 3 96 well plates to be processed in 90 minutes. The DNA extraction was followed by library preparation, and this was mostly automated using biomech robots again. The only part that could not be automated is the adding of one particular reagent, which is quite viscous, and this couldn't really be done on the robot with a high degree of accuracy. So we would add that reagent manually. But overall, there's improved consistency between samples, and it removes potential sources of error. So we did some analysis, and we looked at sequence quality between libraries prepared either on the robot, which is in blue, or libraries prepared manually, which is in pink. We compared 2 different sequencing quality parameters. So we've got uniformity on the left, and as you can see, samples prepared either manually or on the robot, there wasn't really much difference in terms of uniformity. There was a significant difference in terms of reads on target though. So libraries prepared on the robot, there was a higher percentage of reads on target than libraries prepared manually. So next was loading the chip. So we load the chip on the Iron Chef. So this is about 15 minutes hands-on time, and then we leave it to run overnight. The Iron Chef can do 2 chips simultaneously. So you can do 96 samples per 540 chip, which means that you can do 192 samples per run. The S5 prime, so this is the sequencer, this also involves less than 15 minutes hands-on time. Takes about 3 hours to run per chip, although runtime and analysis is 6.5 hours, and then we do our high throughput bioinformatics analysis pipeline. So we designed and validated a targeted AmpliSeq panel. So our gene panel has got 5 genes on it, which are all associated with current newborn screening disorders. The panel was designed to cover these exons, plus or minus 5 base pairs, and overall there was really good coverage, except for parts of the CFTR exon 1 and the TSHR region. So the graph at the bottom on the x-axis is region, so region name, and then in grey this indicates over 100 times coverage. So as you can see, this really shows that we've got quite good coverage. The green is between 50 times and 100 times coverage, which is okay in a diagnostic setting. The yellow is between 30 times and 50 times coverage, so this is on the low side, and as you can see, we've got some of this in our CFTR region. And then the red is the less than 30 times coverage, and as you can see, there's a little bit of the TSHR exon 10, which is less than 30. So again, we did some comparisons. So we compared sequence quality from dry blood spots versus venous blood. And again, we compared, so these 3 sequencing quality parameters, again, percentage of reads on target, uniformity, and percentage of bases with 50 times coverage. And as you can see, sequences from dry blood spots were comparable to sequences from venous blood. So there's no significant difference in terms of sequence quality, which was good. We also had 33 paired samples. So we had samples from 33 patients where we had a dry blood spot sample and a venous blood sample, and we could compare variant calling concordance. So in our 33 paired samples, overall there was 96% mean concordance. However, if you only look at regions with more than 30 times coverage, then this concordance increases to 98%. So overall, we did 7 high throughput runs. So we did 4 high throughput runs on the S5, and the results are shown by the 4 graphs along the top. And we did 3 high throughput runs on the S5 prime, and these are shown in the 3 graphs on the bottom. So again, we're just looking at sequencing quality metrics. So on the x-axis, we have mapped reads, the number of mapped reads. And on the y-axis, we have the percentage of bases with 50 times coverage. Each dot represents one of the dry blood spots, so one of the 96 dry blood spots on our 96 well plate. And we put some thresholds on the graph as well. So the red line is 90% of bases with 50 times coverage. The blue line is 97% of bases with 50 times coverage. And the green line is 99.5% bases with 50 times coverage. And as you can see, most samples meet the threshold of having at least 90% of bases with 50 times coverage. Samples below this threshold have definitely failed, so there's too many gaps in the sequences, and you definitely need to repeat those. You can see that our best results are on the S5 prime, although this doesn't really reflect a difference between models because they use the same chemistry. I think this improvement in terms of consistency and results is really just reflecting our lab becoming more familiar and well practiced in the protocol. But it's quite nice that by the end we've got, by the very end we only had 1 sample sort of borderline below 90% of bases with 50 times coverage. So a bioinformatics pipeline was developed in-house. So these are all the steps in the bioinformatics pipeline. The 4 steps in the middle were automated so that it only involved a couple of clicks. So our genetic technologists in the lab can use this really nice interface where all they have to do is put in the number, the work list number of the NGS run they've just done, and the machine that they used, and click Preview Work List. And then we've got a sort of pipeline progress page and QC steps. So you can see if your pipeline is queued, which would be indicated in grey, if it's running, which is indicated in green, whether the pipeline's passed, which is in blue, and also you can get a warning sign as well, which is in orange. So this just highlights that there might be some things for you to go and look at in a little bit more detail. So we did a bit of a costing model for our high throughput pipeline, and this includes sample reception, so the costs of receiving the sample, booking it onto our lab information management system, dry blood spot punching, dry blood spot DNA extraction, the whole library prep reagents and chips, and also the service contracts for the robots and the Iron Chef and S5 Prime. So these costs do not include our trust overheads or clinical scientist analysis and reporting time. We didn't really look at clinical scientist analysis and reporting within this diagnostic workflow. It was a little bit outside of the scope of our research project. So as you can see, 96 samples with labour is about £69, and without labour it's about £67. But taking into account about a failure rate of about 3 samples per run, then this would increase the cost to about £71 per sample. So this is the current timeline for our high throughput protocol. So on day 1, you can book on the samples, punch them, perform DNA extraction, and start the AmpliSeq library prep. On day 2, you can finish the library prep and do QC using the tape station. You can pull the libraries and load the Iron Chef, which will run overnight. So that on day 3, you can sequence 2 chips on day 3, one after the other. And this leaves day 4 for data analysis and reporting. You can run 2 96 plates simultaneously, and over a 7-day working week, this would mean that you could analyse 1,000 samples per week. This is just limited by your machinery, so if you double up on your automation equipment, then you can increase your sample high throughput capability even further. So I'm just going to pass you on to my colleague who's going to talk to you about a cystic fibrosis screening project that we're currently piloting at the moment in our lab.
Okay, hi, I've just got 2 slides just to finish up on the cystic fibrosis project. So currently CF screening in the UK involves a biochemical test which acts as a pre-screen. Any patients that have a significant result for that biochemical test are then moved on to a genetic test. Currently that genetic test involves a SNP array. So first of all, it's a 4 mutation SNP array and then a 50 mutation SNP array. So at most, using the current method, we're only looking for about 54 mutations. So the problem is that we know that there are more than 300 mutations that are involved in cystic fibrosis. So by only looking at around 50, we're not actually getting the full spectrum of mutations, and we could be missing things. So we wanted to see if, using the knowledge that we had from the newborn screening project, we could expand testing for cystic fibrosis to see if we could identify the other 250 mutations. But we have to keep within quite tight constraints of the newborn screening program. So there's quite a few considerations. First of all, the DNA extraction and the sample type. Again, we're using the dry blood spots, like in the newborn screening project, so there's issues with the extraction and the DNA quality that we get out. The second point is the turnaround time. So again, we have a really tight turnaround time of just 3 working days. So we receive the samples in the lab on Thursday, and the reports have to be out on Monday. So we actually have a lab technician who comes in on Saturday to basically start the sequencing off so that we have the whole weekend to do that. So when we all come in on Monday, the data's ready for bioinformatic analysis and reporting. And that's very quick, actually. Interpretation is, there's literally no interpretation, because what we do is we are looking for hotspot variant calling. So we are only looking for the variants that we know, so only those 300 variants that we know already are associated with cystic fibrosis. And we already know everything about these mutations, so we know how severe they are, we know how they affect the patient. So there's no variants of uncertain significance that we're finding, so no further interpretation is needed, which makes reporting very quick. We don't want to identify carriers except carriers of the 4 most common mutations, and that's just a derivative of the SNP array method, that they still identify carriers of the 4 most common mutations, but the newborn screening committee don't want to identify any other carriers. So we can't just simply show all of the results of the pipeline, we have to hide certain things because they don't want to know. The mutations also have differing severity, so we only want to report patients if they're actually clinically affected. So even if a patient has 2 mutations, if they're both very mild, the patient won't actually have very severe symptoms, they could actually be asymptomatic. So we don't want to report those either because they won't actually need treatment or medicalisation. So this is basically the results that the scientists see on the left as soon as the bioinformatic pipeline is done. So the pipeline is basically the same as the newborn screening pipeline that Rebecca showed earlier. We use the results of the pipeline to classify the patients into different categories. So the first category is they're affected. They've got 2 mutations and at least one of them is severe, so they are clinically affected with cystic fibrosis. And that's the red button there in the table. The second category is the carrier report. So these are the carriers of one of the 4 most common mutations that they want to know about. So those are the carriers that we're reporting. And then the last category is what we call no significant genotype. So that's either patients who have no mutations found, or they have 1 mutation that's not one of the 4 most common ones. So they're a carrier, but we want to hide them. So the scientists can only go in to look at the results of the first 2 categories, so only the ones that they're reporting. They can't go in to look at the results of any patients that have no significant genotype, so we actually lock them away from that data in case they stumble upon a carrier by accident. But they can see that the sample has passed QC, so we know it's a good quality sample. So basically the overall results of running this panel, these are actually a month out of date, so these numbers have gone up a little bit. But at the time of writing, we had been running the panel for 9 months. We had just over 200 samples in total. We had reported 14 carriers, so those are the carriers of one of the 4 most common mutations. And we had 21 affected newborns. And importantly, actually, 7 of those wouldn't have had a genetic diagnosis without the NGS method. So they either had 1 or 2 mutations that were not in the SNP array panel, so we were finding their second mutation. I think this is actually 9 now, so 9 of them wouldn't have had a genetic diagnosis without NGS. We can also detect CMVs as well. So we're using a panel that was a community AmpliSeq designed panel. So it's different from the MBS panel, it's just CFTR gene. But actually the known breakpoints for some common CMVs were designed into the panel, so they're completely intronic regions, but they're in the panel. So they're sort of slap bang across a breakpoint of CMVs. So we can detect the reads are split and they start getting clipped if there's a CMV present, so we can also pick up CMVs. We've had all the samples reported on time, apart from 1 run. So all the samples failed, so that's a problem that hasn't happened since, so hopefully we can improve on that. So overall, we've still got 2 months of this pilot project, so we're still going to be running it for a bit longer. And after that, then it's going to sort of be up to the newborn screening committee to see whether we actually carry on with this method. But hopefully they'll see that it's a bit beneficial over the current SNP array method, so they'll try and use it. So just to finish up, on behalf of Becca and myself, we'd just like to acknowledge all the people who have helped us with this project, so all of the Sheffield team, past and present, who have helped us. A lot of other NHS trusts in the UK are collaborating with us on this project as well, and also, of course, Thermo Fisher for the good collaboration too. So thank you.